This is a lecture on genetics from October 12th, 2020. And in this part of the lecture, we're going to define some terms that are going to be important in understanding genetics, as well as talk about how to solve genetics problems using Punnett squares, as well as the probability method. And so when we talk about genetics, we have to sort of start with the father of genetics, or Gregor Mendel. And Mendel is an Austrian monk who bred and studied the principles of inheritance in pea plants, which you can see down here. And pea plants are a great model to study inheritance because they self-fertilize, which leads to very little variation between the peas from generation to generation. And it allowed Mendel to really identify specific traits within these peas and study them. And it also allowed him to generate what's known as true breeding, or kind of purebred strains of peas, where every single offspring would have the same traits as the parents. And so a true breeding pea plant that has pink flowers will only ever have offspring with pink flowers as well, because it's a purebred pink plant, and that means its offspring are all going to look the same as the parent. And so Mendel started to wonder what would happen if you kind of didn't allow pea plants to self-fertilize, but rather cross-fertilize them with each other. And you took one true breeding plant with one trait and crossed it with another true breeding plant that had a different trait. In this example here, the true breeding trait we're talking about is height, right? And in this case, on the left, these would be the true breeding tall plants. And they were crossed with true breeding small plants, or dwarf plants. And then once Mendel did this cross-fertilization of a tall and a dwarf plant, he looked to see what kind of progeny they generated. And this is what's known as a monohybrid cross, because we're studying one specific trait. We're studying the height of the pea plants, right? And we're looking, and some pea plants are tall and some are dwarf, right? And Mendel crossed a tall and a dwarf plant, and what he noticed was that every single one of the progeny of this tall and dwarf plant were all tall. So the short plants, or the dwarf plants, seemed to disappear in that first generation. But if he took these progeny, these tall hybrid plants, and allowed them to self-fertilize, hence like basically one tall plant progeny crossed with another tall plant, in the next generation, he noticed that the short plants reappeared. And there were still some tall plants. There was actually about 3 times as many tall as there were short. But it seemed like the short factor, this dwarf factor, was masked by the tall one, and then reappeared in the second generation. And so from his monohybrid crosses, or his crosses studying one specific trait, Mendel came up with a series of terms to describe the relationships that he saw. And the first term that he really used was gene, right? And genes are just heritable units or traits. So in the previous example, that would have been height, right? And alleles are different forms of genes, or different forms of that specific trait. So the gene from the previous example would have been height. The alleles would have been tall and dwarf, different forms of height, right? Another example is here. Eye color is encoded by a gene, and there are 2 forms or alleles of this gene. There's a brown allele for eye color and a blue allele. And the allele, or the form of the gene whose expression is favored, is known as dominant. And the one whose expression is hidden is recessive. So in the previous example, since the short plants were the ones with the hidden, that were hidden in that first generation, the short or dwarf allele is recessive, whereas the tall allele was dominant, because that was the one whose expression was favored, the one which we saw. In the case of eyes, brown is actually dominant to blue. And usually we express dominance and recessiveness as capital letters for dominant and small letters for recessive. And since we are diploid organisms, and as are most other organisms, we have 2 alleles for every single gene. And sometimes those alleles are both the same. And that would make the individual homozygous for that particular gene. And you can see here this would be 2 big B alleles or 2 small b alleles. But as long as these 2 are the same, that individual would be considered homozygous. Same, right? If the individual has 2 different alleles for one gene or 2 different forms of one gene, they would be known as heterozygous, right? Because hetero is different. And then you can see an example of that here, where that individual would be big B and small b. And when we're talking about these big B's and small b's, what we're really talking about is the genotype, or the alleles for each gene, or the genetic makeup for each gene. But when we're talking about what we actually see physically, that's what's known as a phenotype. So phenotype is the physical characteristics that we see. And so an example here for the eye color, we have a homozygous individual, small b, and that small b would be considered the genotype. And since we know that small b would be a blue eye and blue eye, this homozygous recessive individual has a blue eyes phenotype, because we see the blue eyes and their genes are BB, right? So small b is the genotype, blue eyes are the phenotype. And you can basically create a table that lists all the possible genotypes resulting from any genetic cross, or any monohybrid cross in this case, by listing all the possible sperm genotypes on one side and all the possible egg genotypes on the other. And then you put the possible genotypes of each zygote, or combinations of the sperm and egg, inside the 4 squares. And this whole table is known as a Punnett square. So you have 2 parents with 2 genotypes for ear lobes, in this case, where big E is dominant over little e, and big E is unattached earlobes, and little e is attached earlobes. And so you could see here, one parent, this is the dad, has a genotype of big E, small e, so he's heterozygous. And so you put that big E here, small e here, and that would be dad's genotype. Mom is also heterozygous, so you would put a big E, small e on the top of the Punnett square here for mom. And then, in order to make zygotes, or possible kind of offspring for these 2 parents, you would take the big E from here and the big E from here and put them both into this Punnett square to make a homozygous dominant individual who has unattached earlobes. And you would do the same here. Big E and small e would make a heterozygous individual. In this square, small e and big E make a heterozygous individual as well. All 3 of these have unattached earlobes, because unattached is dominant over attached. And this individual, because they have a small e and a small e, or homozygous recessive for this trait, they have attached earlobes. And so what Punnett squares really allow you to do is calculate the probability of both the genotypes, big E, little e, for example, as well as the phenotypes associated with the offspring of 2 parents. And so we're going to go through this example, show you how another way that can be done. And so in mice, if we have 2 colors of mice, some with black fur and some with white fur, and black fur is dominant over white fur, and then we take a black mouse that is big B, small b, and mate them to a white mouse that is small b, what is the percentage that their offspring will be white? Okay, so first, you can list out the genotypes of the parents. We have a black, big B, small b, heterozygous mouse, right? And a white, homozygous recessive mouse, 2 small b's. And you can set up the Punnett square with the black mouse on the top, big B, small b, and then the white parent going down the side, small b, small b. And then if you fill out the potential offspring genotypes in the center, you have a big B, small b, big B, small b, and then 2 individuals who are small b, small b. And the question asks what the percentage of offspring that will be white. And we know that white is recessive to black, and in order for white to be seen, we need to have 2 copies of the white allele, or be homozygous recessive in our genotype. And both this individual here and this one are homozygous recessive. And that means that 2 out of 4 of the offspring will be white, or 50%. So what if we change the genotype of the mouse to be homozygous dominant rather than heterozygous? And it now is a black mouse with a big B genotype crossed to a white mouse, which is small b, small b. How many of their offspring will be white then? So if our parent is big B, black mouse, times white, small b, we can have 2 big B's for that parent on the top, 2 small b's on down the side. And then all of the individual offspring will have the same genotype. Big B, small b. They'll all be heterozygous. And since we know that in order to be white, you have to be homozygous with 2 small b alleles, none of these offspring are going to be white, right? Because they are all heterozygous, which means they are all going to be black, because black is completely dominant over white. And so it's easy to know the genotype of an individual who shows the recessive phenotype. Because in order to show the recessive phenotype, you know you have to have 2 copies of the recessive allele or be homozygous recessive. But if you think back to those 2 previous examples, how would we determine if an individual that has the dominant phenotype, like the black mouse, is homozygous dominant, has 2 big B alleles, or if they're heterozygous, has a big B and a small b allele, because they look pretty much exactly the same. And so how do we know the genotype of an individual who has the dominant phenotype? The answer is we can do what's called a test cross. And a test cross takes the organism with a dominant phenotype, like the black mouse from the previous example, and crosses it with a recessive individual, or white mouse, like that previous example. And just like we did in those 2 crosses, what you would notice is that if the dominant phenotype mouse is homozygous dominant, 100% of the offspring resulting from this test cross will also have the dominant phenotype. So a big B mouse crossed with a white mouse will result in all heterozygous offspring, therefore all dominant phenotypes, right? All black mice. But if you have a big B, small b mouse, or a heterozygous individual, then 50% of the offspring from the test cross will be dominant phenotype, and 50% will be recessive. Because we have a big B, small b crossed with a 2 small b's of a white mouse, for example, we would end up with 50% heterozygous offspring and 50% homozygous recessive, meaning that we'll see 50% of those individuals having the dominant phenotype and 50% having the recessive. And so Mendel ultimately came up with 2 important principles. His principle of dominance, where there's one allele or form of a gene that can conceal the presence of another, where dominant masks recessive, and his principle of segregation, where each individual has 2 alleles or 2 forms of each gene. And these alleles segregate during formation of gametes during meiosis, so each gamete only has 1 allele for each gene.